So this section is going to be all about the ways that people have tried to make pox viruses more useful, and one way in particular that ended up making pox virus more dangerous. So this is an interesting little ethical dilemma, and also an interesting scientific one. So yeah, take out my gum. There we go. Let's go. All right. So this happened a while ago, but it's a classic. So let's go through it. So 2001, scientists unintentionally engineered a virus that becomes fatal. And the weird thing is that this is fatal even in mice that have genetic resistance or that have been vaccinated. And the thing that they were modifying was a vaccine strain of a virus, so a live attenuated virus that was going to be used as a vaccine. And so this sounds horrible, yeah. And so what do if you get this result? Well, the answer is, there's a lot of answers, but you could go to the New Scientist and write an article and give them a quote. Disaster in the making. An engineered mouse virus leaves us one step away from the ultimate bioweapon. Oh my goodness, yeah. And so one of the heads of the project, Dr. Jackson, said that we wanted to warn the general population that this potentially dangerous technology is available. And we wanted to make it clear that the scientific community should be careful, that it's not too difficult to create severe organisms, which is actually quite difficult. But they got lucky, and so they thought it was pretty easy. Yeah. And time since then has shown that it is particularly difficult to make a virus or something any more dangerous than it already is. Thing was, this happened in 2001. And if they'd actually gone and done their homework and read some papers, what they would have found is that, British and Australian scientists had produced or previously published similar results. So the way, though, that they did it is quite different. So the title of this paper was IL4 Insertion in a Modified Vaccinia Virus. And so that's the only title. The title was not Disaster in the Making, how to engineer your own ultimate bioweapon, essentially. And tucked away right down near the end of it was that, mortality, like death rate of 100% was observed for mice that were immunized with this virus that had a modified gene, so it had an extra interleukin number 4 gene. And this contrasted with the death rate for mice that were immunized with regular vaccinia virus, which showed no ill effects. So vaccinated mice were fine, but if you put in this particular interleukin 4 gene, that seemed to cause the virus to break through any resistance and start killing the mice. And this was back in 92, and then the other one is in 98. Or rather, this was the 98. The 92 is down here, so it had been done many years before. And so the title of this one is Recombinant Vaccinia Virus Co-expressing the F protein of respiratory sensitive blah, 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 interleukin number 4 does not inhibit the development of memory T cells, whereas priming is diminished in the presence of high levels of IL-2 or gamma interferon. So there's nothing about either of those titles that, imagine you're sitting in your terrorist cave reading, Terrorist Weekly magazine. Which one of these things is going to make you sit up and take a sip of your coffee? Yeah, I think it's going to be the top one. Yeah. So these are all scientists that are essentially doing the same thing or doing very similar experiments and getting the same conclusions and reporting them in very different ways. Or rather, one of these things is not like the other ones. One of these things, Dr. R. Jackson, does not belong. All right, so what's going on here? All right, first of all, interleukins. There are a lot of interleukins in the world, and they do a lot of important things. So interleukin 1, alpha and beta, but particularly beta, is going to be pro-inflammatory. So it's going to drive an inflammatory response. Interleukin 6 is going to be a thing that kind of starts off an inflammatory response. So an inflammatory response is when basically macrophages are going to get involved and just start smashing the place up. That's a real simplified version of it. All right, so yeah, that's great. And you got these macrophages that are talking to each other and talking to fibroblasts and talking to other cells in the immune system. And interleukins are like interleukin. Leukocyte means white blood cell. And interleukin is for communications in between, the white blood cells. That's the entire idea right there. And so they're little messages that cells in the immune system can pass to each other to figure out what to do about a situation, and so that the entire immune system is on the same page with respect to how it's going to respond. And over here, I think this is going to be a T cell. I think that's what we've got on there. All right. So what does interleukin number 4 do? So interleukin number 4 is the gene that they put into a pox virus. So it was a regular pox virus. In this case, it was a mouse pox virus called ectromelia virus, which is just the name for the mouse pox virus. And it's going to be equivalent to smallpox, but it's going to work in mice. It doesn't cause that same level of horrendous disease. It's actually quite mild. It's just a genetic relative. So they made a version of this that, in addition to all of its normal genes, it also had an extra gene for the interleukin 4 of a mouse. And so when the virus was there, it would make extra interleukin 4. And the idea was that they could then use this to make vaccines. So that's why all these groups were trying this same thing. They were all trying to make better vaccines. And there have been a lot of people trying to make poxvirus vaccines for a lot of years. Yeah. And there have been a couple that have gone through clinical trials, but there hasn't been anything, as far as I'm aware, that has really been a home run yet. But it's something that people are, they keep trying. Because pox viruses can go into your cells, they can replicate, they're fairly mild, your immune system can control them, and they're huge and made of DNA. And so you can put as many genes as you want into them, and the pox virus will act like a little truck and take those genes to a cell and then express them for you, which is a great way to think about starting a vaccine, essentially. So the idea here is that they wanted to drive a vaccine that made really good antibodies. And so they wanted to make this interleukin number 4. Because it would essentially drive B cell production and force them to turn into plasma cells and memory B cells. And these are things that are going to make antibodies, and so you get an antibody response. That's the idea. So the way IL4, interleukin 4, works is it's going to interact with a particular population of T cells. So in this world, there are helper T cells and killer T cells. We're going to focus mostly on the helper T cells. So the helper T cells have a protein on the outside called CD4, and this is the same one as one of the attachment receptors for HIV. And so that's why HIV hits helper T cells. It's looking for this particular protein, and they are the cells that have it. And so there are actually 2 populations of these helper T cells. They start out as baby helper T cells, and we call those T helper type 0. So type 0 means they haven't really decided what they're going to do with their lives yet at this point. And if they get a little bit of an interleukin 2 signal, then these helper T cells will start to divide. And so, okay, that's great. And then if they get no signal, they will turn into helper T cell type 1. So we call these Th1 cells. And Th1 helper T cells are going to go out and present antigens to killer T cells. And they're also going to make memory T cells and put those away. So these guys are going to drive a T cell mediated response. They're going to go in and support macrophages and killer T cells going about their work. And so we call that whole thing a Th1 type response, which is going to be mainly cytotoxic T cells, those CD8 T cells, killer T cells. And so you can see the helper T cells are not directly doing that. They're going to release signals like interferon gamma that are going to bring T cells to the right place and are going to help control how macrophages work. Macrophages are they're the things that cause most of the disease in Ebola and avian bird flu and in SARS coronavirus 2, for example. They're just another message. Okay. So interleukin-2, when these cells receive it, once again, that's going to make them divide, and so that's just a general signal. When any of these T cells gets interleukin-2, it's going to say, let's divide, let's make more of ourselves. But interleukin-4 is the key here. So when we get interleukin-4, what it's going to do is, interleukin-4 is going to send us from a Th0 cell to a Th2 type cell. And so Th2 cells are going to start to present antigens to B cells. And B cells, if they are successful at making something that matches the antigen, will get to turn into plasma cells and memory B cells, and the plasma cells will make antibodies. So this is the start of the process to make antibodies. B cells don't directly make antibodies. They make a thing that looks like an antibody, but it's hooked onto the surface of the cell and kind of embedded there. And then if that gets a really good match to whatever the T cell is holding out, then they get to differentiate and they become plasma cells. So that's great. And so IL4 drives that. And so they said, if we put in a virus and we've got IL4 there, then we are going to drive a B cell type response and just focus the immune system's attention on making lots and lots of antibodies, which is true. Because they also knew that interleukin-4 would inhibit. So here a red arrow like this one or this one is going to mean inhibits, and a green arrow like all the other arrows that are a color over here, that's going to mean promotes. Yeah, that's bad for red-green color blindness, but yeah, I suppose I need to redesign this slide at some point. I stole it from somebody, I'm sure. Anyway, so B cells are actually going to make interleukin-4, and when they do that, they're going to shut down this Th1 type response, which is going to be making memory T cells and sort of support helper T cells that are going to support a killer T cell type response. And so that seems all right? That seems like it makes sense. You want to make more antibodies? Fine. Make one gene, IL4, and it turns on more antibody production and less T cell production so that the immune system is totally focused on making antibodies. Pretty cool. Problem was that this was a live virus. And when they did this, it turns out that this live virus doesn't normally kill anybody. But in this case, it became 100% lethal. And that was the problem. So let's see. Over here, we can see, all right. So these are two different types of mice. BALB-C mice are those little white mice. C57 Black 6 is what that stands for. These are the cheapest kind of black mouse. These are both inbred lines, and they're both pretty darn cheap. You can buy them for, I don't know, $2-3 a mouse. And then it costs something like $1 a day to feed them and house them and clothe them in the style to which they're accustomed. It might even be more than that. Rent tends to go up. Been a while since I did anything like this. Anyway, what they're looking for here is the response of mice to reinfection. So if they were already immune to ectromelia virus, so the mousepox virus, they'd been given mousepox, they got over it, they had immunity built up. Then when you reinfected them with mousepox that had interleukin number 4 gene in it, now this second wave is going to go in and it's going to turn off all the Th1 response, all of the killer T cells, and all of the support for the killer T cells is going to be off. And we're just going to leave the antibody response on. And what happens is that when they just put in the regular virus, again, with no IL4, none of the mice die. This is the mortality at 6 to 8 days, so like a week after. And there's 5 mice in each group. But when they put in the IL4 version of the virus, then all of a sudden, the mice start dying. So this is 60% in this particular one that are dying. And depending how you do the experiment, you can get somewhere between about 60%, this is a good outcome, and 100% of the mice that then die. This is worrying. Imagine, so right now you've probably had a polio vaccine, you probably had a number of vaccines. Imagine there was a virus that could make your immune system forget that you'd been vaccinated. Then all of a sudden, you would be susceptible to polio or reinfection of any kind. That's basically what they've done with this IL4 virus. When they put it in there, it made the mouse forget that it was already immune to this virus, and the immunity didn't work. It was blocked by all of the IL4 that was made. And ectromelia virus is not ordinarily fatal in black 6 or Balb C mice. They normally get over it pretty quickly, but not this time. Okay, and so the question was why? What's going on here? What's different? So in mice that were treated with just the regular virus, so they were immune and then given the regular virus again, this is what their T cells were doing. So this is a T cell killing assay. And basically, if the numbers are up here, we're going to dilute the sample. So we would expect the numbers to go down, but they should follow a nice sort of linear path. And that's just to make sure that we're measuring what we think we're measuring. And so you should see lots of killing up here. And so we're destroying somewhere around 25 to 35% of those cells. And that's pretty good because these cells will get to work for longer inside of a mouse, and so they can actually kill 100% of the cells over time. So that's what the immune system should have looked like and did look like in the mice with the regular virus. And with the IL4 virus, this is what you saw, which was no T cell killing of any sort at any concentration. So those killer T cells, those cytotoxic T cells, were absolutely switched off. Now, NK cells, natural killer cells, which you'll remember from some of our previous viruses that we looked at, like, for example, the Ebola story, those are only slightly reduced. So there's other figures in this paper that we're not going to go through. And they mostly work as normal. But cytotoxic T cells, CD8 positive T cells, do not actually make any specific response. And this is because of the IL4 there. And so this virus, which is supposed to be like a vaccine, it's supposed to go in, your immune system controls it, and you get better, or the mouse gets better, now becomes lethal because they put in a gene from our own immune system. So yeah, that sounds bad, right? It is pretty bad. Okay, so then people tried this out with other things, and it turned out that you got similar phenomena with vaccinia virus. And vaccinia virus is the vaccine for smallpox. These experiments were done in mice, of course, not in humans, and it was with the mouse interleukin-4. And we don't know if the human interleukin-4 is going to work in exactly the same amount, or it should be the same general pattern, but yeah, it's questionable. And so with vaccinia virus, once again, the amount of NK, natural killer cells, is about the same in normal, which is going to be black, and interleukin-4 virus-infected situations. So yeah, they're about right. But look at this. The amount of virus that stays is just going to crash down in the normal infection. So normally this would be completely controlled in the mouse by about 12 days and drops down below the level where we can detect it. But the IL4 virus is allowed to linger. Look at that. And so we turn a 2-week infection into a 3-week infection. We go right out to almost 22 days right there with virus positive. And that's because IL4 is shutting down the T cell response. And cytotoxic T cells, those killer T cells, and the Th1 response, this is absolutely the key to stopping Ebola virus, coronavirus, influenza virus. Pretty much any virus that you can name is going to be controlled by killer T cells. The Th2 response, the antibodies, can prevent you from getting infected in the first place. But that's about all they're good for. They're usually not enough by themselves to actually stop you from getting sick if you do get infected. The role of stopping you getting sick is all about those killer T cells. And shown here, this is what happens to the killer T cells. So these are two different types of assays. And this is the normal response in black in both cases. You can see a very nice high response. And here's the response when we have that virus that is making its own interleukin-4. So it's a virus that's basically making its own camouflage. And this is something that people did with good intentions, but on purpose. So yeah, this is, I don't know, if you're talking about weaponizing or just ways that you can accidentally make the world worse with a virus, this would be one of them. This is a real good example of that. Now, this story actually isn't quite as bad as it seemed. So there's a nice big review about this, which is where I stole some of these figures from. And the idea is that, yes, in the mouse with ectromelia virus, this is particularly severe. But in the mouse also with vaccinia virus making mouse interleukin-4, it's not as severe. So vaccination, it can't break through vaccination with the vaccinia. And also they did this with a rabbit pox virus. It's called myxoma virus. And this grows in rabbits. And this also does not break through vaccination. So it was only that one case with a mouse with interleukin-4 that we actually got the vaccine breakthrough. And so that means that probably something like this is not going to be an amazing bioweapon. It's probably not going to take over the world, thank goodness. And that's actually really good, right? This is, this is not what we want from our science. So interleukin-4 expressing viruses may not be safe, but it does look as though it would probably be possible to control them by vaccinating. And if you've been in the military, you know that we still have and use, particularly in the military, smallpox vaccines. So that would be the thing. If you were vaccinated, you would have a little scar about the size of a quarter, and it's going to be on one of the muscles up by your shoulder. That's typically where they do that. And they do that so that if the enemy ever decides to release bioweapons like smallpox, then they just do nothing. And so that just prevents anybody else from even trying to do that particular kind of biowarfare. Now, the other interesting factor is that pox viruses actually replicate inside your cell like all these other viruses. They're going to replicate in the cytoplasm. But when they're in the cytoplasm, they have access to all those copies of RNA, anything that the cell is making. And from time to time, there will be a retrovirus that gets activated in the cell. And remember, we're all built mostly of copies of little retrovirus pieces, things like HIV from back in caveman times. And so from time to time, one of those reverse transcriptases gets turned on. And we don't really know why it happens. Probably there's a different cause every single time it happens, but sometimes it does happen. And so that reverse transcriptase would turn potentially regular messenger RNA into a piece of double-stranded DNA, which it does. And then if that happens, vaccinia virus and other pox viruses seem to be very good at picking up these genes. And we know this because these are some of the genes that pox viruses have picked up. They are masters of immune evasion, and you could run an entire course just on the way that pox viruses shut down the immune system. But essentially, these are all, so everything in gray up here is something that the cell could make that would actually help it. So there's interleukin-2 that's going to make T cells proliferate. Here is interleukin-1 beta that is going to be a pro-inflammatory cytokine. It's going to activate a bunch of other things. Here's interferons, the interferon gamma. So this is for communication among those white blood cells. Interferon alpha, this is the regular interferon response that comes from that RIG-I MDA5 pathway. And there's so many other things down here. There's your CC chemokines and your CXC chemokines. And you'll know those as the receptors for those as things that are involved in HIV. And in each case, vaccinia virus makes something that looks like the normal human receptor, but it doesn't work. So normally a receptor will bind to a protein and will then send a message. What this entire column is is these are soluble receptors. So a receptor is supposed to be hooked up to something else. So when it binds interleukin-1 beta, then you get signals that happen inside the cell, and the cell starts to change. In this case, that doesn't happen because we've just sent out something that binds to interleukin-1 beta and is connected to nothing. And poxviruses don't just do that the once. They will send out little tiny decoy receptors, and they will grab all of these different factors that help the immune system to function. And poxviruses are doing this so that they can avoid detection by the immune system. Now, poxviruses also have another couple of things. So they have proteins that will bind to regular human receptors. So the poxvirus is actually going to do some signaling. So for example, interleukin 10 right over here is something that'll turn off an inflammatory response. And so poxvirus is going to grab the messenger that would turn on an inflammatory response and stop it. TNF alpha over here is also another thing that will cause an inflammatory response. And it's going to make an extra copy of, like a fake viral copy of a basically all clear signal, which is what interleukin 10 does for the body. And so, yeah, it's saying, oh, there's nothing to see here. Just, I don't know. Go pay attention to something else. Immune system. Yeah, this cell is not infected. That's for sure. We know that poxviruses also make some of these 7 transmembrane chemokine receptors of their own. And these are going to be receptors that bind to some of these chemokines, the CC and CXC chemokines probably, and are probably not going to have the intended signal. So and just decoy after decoy for things inside the cells and outside the cells and communication in the immune system. Poxviruses take a human version of a gene, modify it, take away some part of it that actually makes it work, but still allows it to bind, and then just send these things out into the world. So yeah, that's part of how they do it. And the fact that poxviruses have interleukin 10 analogs, and they have mimics that can bind to interleukin 1 and 2 and 5 and 18. This suggests that at some point in history, poxviruses probably had the chance to get their own copy of interleukin 4 because they've been around cells. I mean, that's the one place where they grow. And they have sampled very heavily from their hosts. They've stolen all these other interleukins and interleukin receptors from them. And so the fact that we don't have interleukin 4 viruses actually naturally in the world suggests that there is probably not an advantage for the virus. At some point, this has almost certainly happened because we know that, for example, making interleukin 10 is something that several different groups of poxviruses have independently picked up this ability from their hosts. So it seems to be something that happens relatively quickly after a virus gets into its host. And IL4 just doesn't get there, which means that overall, this virus is better off not making interleukin 4, which again is probably positive news. This, from the virus's point of view, it probably just squeezes out a few more copies of virus if it is not actually killing its host right away. But from your point of view, this means that killer viruses will actually get outcompeted by non-killer viruses in the world. And so once again, it's one of those cases where the other viruses are actually going to keep you safe from some of the worst viruses. And yeah, in a way, I find this comforting. Competition is actually our friend. Competition between viruses is very helpful and very protective. So there we go. It's a weird little story about interleukin 4 and poxviruses. And have a look at that and get studied up on that for the test question would be my recommendation. Thanks very much.